So, hello everyone. Now, in continuation with what I discussed in the electron transport chain and oxidative phosphorylation, I give an introduction about the electron transport chain, where it is positioned and where these complexes are arranged. So, this was the thing that I was talking about. So, it is the all the complexes, that is complex 1, 2, 3, and 4. They are situated in the inner membrane, and we have two mobile carriers, that is ubiquinone and cytochrome C. Now discussing more in detail about how the electrons are being transferred. So here we see this upon oxidation of NADH to NAD. So the electrons are accepted by some few electron carriers. They're given to ubiquinone, and ubiquinone give the electron from complex 1 to complex 3, and from complex 3 they are being taken up by cytochrome C, that again is a mobile carrier, and given to complex 4. So while this process is taking place, that is the transfer of electrons, we see H ions are being transferred from the matrix of the mitochondria to the inner membrane space. And similarly, when FADH is oxidized by complex 2, then again the electrons are taken up by ubiquinone, that is a mobile carrier, and given to complex 3. Complex 3 gives the electron to cytochrome C, and then to this complex 4, and final electron acceptor is our oxygen, which is reduced to generate H2O. So while this process is taking place, we see a number of H ions are being transported from the matrix of the mitochondria to the inner membrane space. So here we can also see, like with the oxidation of one NADH molecule, we see this 4 plus 4 plus 2, that is 10 H ions are transported, whereas with the oxidation of FADH, 4 plus 2, that is 6 H ions are being transported. And this we are going to discuss in detail while calculating the ATP count of NADH and FADH. So now again more in detail. Here you see complex 1 NADH oxidized to NAD plus. Electron which are being released, they are being taken up by electron carriers, that is FMS, iron sulfur centers, and these electrons are taken up by ubiquinone. And here you also see this red uh, arrow gives the information about the transport of H ions from the matrix of the mitochondria to the inner membrane space. Now this ubiquinone, it has brought up that electron to the complex third, that is cytochrome C oxyreductase, and again the H ion are being transported from the matrix to the inner membrane space. Now this electron is being taken up by cytochrome, and this cytochrome has again brought the electron to the complex 4, and final electron acceptor is our oxygen, which ultimately forms this H2O, and then also two H ions are being transported from matrix of the mitochondria to the inner membrane space. This is the overall view of the transport of electrons and oxidation of NADH molecule. So now discussing the oxidation of FADH molecule. So this, upon oxidation to FAD by succinate dehydrogenase, the electrons are again taken up by some iron sulfur proteins. The electron given to the ubiquinone, and that ubiquinone being a mobile current give the electron to the complex third, that is cytochrome C oxyreductase. Now this electron again taken up by cytochrome C, that again is a mobile carrier, and the cytochrome C will be taking up that electron to the complex 4. So here we see. So we can also conclude from this, while this transport of electron is taking place, there is one more thing that we have discussed, that was a simultaneous process, that is the development of a positive charge in the intermembrane space of the mitochondria. That is the two membrane of the mitochondria, outer membrane and the inner membrane, there is a space, we call it as the intermembrane space, and a lot of H ions are being deposited during this electron transport is going on, and ultimately resulting in a proton motive force because of the high positive charge that has that has ultimately resulted due to this electron transport. And next to the complex 4, here also we see there is a, sometimes some books are saying it as complex 5 or ATP synthase. So when H ions, when they try to again balance the charges, when they move through this ATP synthase, so then they causes the movement in the ATP synthase. So it can be understood by a similar mechanism when a dynamo, when the string of the dynamo is pulled, then that wheel of the dynamo moves. Similarly, that string is similar to the H ions moving from the intermembrane space to again back to the matrix, which causes the movement in the ATP synthase that ultimately result in phosphorylation of the ADP, thereby generating ATP. So here we see there are a lot of H ions are being seen over here in the intermembrane space, and when this H ion movement is taking place, this movement in the ADP synthesis is taking place, which causes the phosphorylation of the ATP, and it is ADP, and it is understood by binding chain mechanism when ADP and phosphate they bind to this cycle, and while this movement is taking place, it is being phosphorylated to generate ATP, and ATP is released from this. So that is the overall mechanism of generation of the ATP via the oxidation of NADH and FADH molecules, which are being generated during the glycolysis step, during the preparatory step, and during the citric acid cycle. So thank you everyone. I'll come up with next video to finally understand the calculation of the ATPs, how much ATP is generated in during NADH oxidation and FADH oxidation. Thank you so much.